Hi class, my name is Alicia Yergi. Today is July 15th, 2021. The time is 10:16 p.m. This is for Bio 225, exercise 2. So we are actually taking the results from the cultures that we grew, and we're going to be plating them today on the agar that we poured in the Petri dishes. So first, what I did to set up, I washed my hands, made sure my area was clean, and wiped down with the bleach solution. Gloves were applied with the rest of my PPE. And again, read instructions a few times before putting everything together. So again, what we're gonna do is we're gonna take our cultures that we grew and plate them on the agar in the Petri dishes and let them grow for 48 hours. And we will result them with pictures and our final results in the data tab for our lab. Okay, so first I have my tubes here that match the Petri dishes that are labeled. So I have my E. coli, the epidermidis, and the yeast. So what I'm going to do is I'm going to take my E. coli first, and and we have our alcohol here with the inoculation loops. So they've just been sitting there sterilizing. So what I'm going to do is I'm going to open the E. coli, and I'm going to place the lid upright in that position, and then I'm going to go ahead and sterilize the top of the tube before I do anything. Okay, so I'm gonna take one of my inoculation loops and shake off the excess alcohol before I place it in the nutrient broth with the E. coli culture. All right, so now that it's sterilized, I'm gonna go ahead and place the loop inside the vial without touching the sides, and kind of stir it up a little bit so I can get some of that bacteria. Okay, I'm going to slowly pull that out. And then I'm going to go ahead and open this up. And I'm going to go ahead and make a zigzag on the agar with the loop. It's almost like a jelly texture in one of the quadrants. Okay, and then I'm going to go ahead and cover that back up and sterilize the loop again. And without putting it back in there, what I'm going to do is I'm going to, again, shake off the excess alcohol from the loop. And then I am going to go ahead and open my agar plate back up. And I'm actually going to drag that initial bacteria that we put in there and zigzag it down into another quadrant. Okay. And I'm going to cover this back up, and then we're going to go ahead and sterilize this again. Okay, and then we're going to shake off the excess alcohol. And then we're going to pull this down into another quadrant again. Okay, and then I'm going to close it back up. And for the last quadrant, I'm going to let this sit for 20 seconds. Okay, then I'm gonna shake off the excess alcohol and place this in the last quadrant, dragging from the previous quadrant that was made. Okay, so I'm gonna take the lid off, place it upright, and then we're gonna drag that okay, and then so that is for the E. coli. I'm gonna go ahead and repeat these steps for the S. epidermidis and the yeast. Again, doing the same steps. Let it sit for 48 hours and see what grows. And then I'm gonna go ahead and sterilize this and get rid of this one and place the cap back on. And that concludes the example of plating the bacteria, and then we'll see what happens in the next 48 hours and we'll post the results. Thanks.